So thank you so much for the invitation. I think it's really a good opportunity for my colleagues here at McGill to deliver such a series of the talk related to the food safety research areas that's going on at McGill. Is pretty exciting. So the title of my presentation today is Novel Devices for Rapid Detection of Food Contamination and Adulteration. Most of the work was supported by NSERC, and also I also want to emphasize that I moved my lab from University of British Columbia to McGill last summer, and most of the works were done at UBC in Vancouver, Canada in the past 7-8 years, and a certain portion of the work has been done here at McGill. So the overall objective for this research program is to develop a novel, rapid, cost-effective, and user-friendly approach for food authentication, therefore protecting all the parties from any potential economic loss and the potential health risks. So food fraud is for sure a very big topic in the food science area right now, and we see there is a very clear connection between food fraud issues and the food safety issues. So that's why we want to develop novel methods for food fraud identification. The current methodology or approach for food authentication is always time-consuming, labor-intensive, and costly. In addition, the sensitivity and accuracy are not fully satisfied. We also require high trained personnel and highly rely on the lab sources. That's why we always ask ourselves, is it possible we can develop other novel detection methods? So the objective is to develop or fabricate rapid, cost-effective, sensitive, accurate, portable, and or user-friendly instrument and sensor-based methods to identify the risks of the food integrity in the agri-food products. So for today, I want to introduce the 3 types of the tools developed in my research group for food authentication study. So the first one is we call the vibrational spectroscopy, including the infrared and the Raman spectroscopy. So they are very rapid, they're user-friendly, and it's portable. So the second tool is called nucleic acid-based analysis. So they can achieve sensitive and accurate detection for different biological species. And the last tool I want to introduce today is called the microfluidic lab on a chip. So this is a very miniaturized device that can be used for portable and high throughput screening for food authentication. So let's move on to the first topic, the vibrational spectroscopy. So both infrared and the Raman spectroscopy belong to this we call the vibrational spectroscopy that offer the fingerprinting features to elucidate the chemical compositions of your food samples, because the vibrational signals of the functional group within the molecule can be recorded as a spectrum. So the methodology is non-destructive, but sometimes it's less destructive. They're very rapid and they're user-friendly. So it's very similar that each human being has our own fingerprint, right? And each molecule, due to their unique chemical composition, they will have their unique fingerprinting spectral features. On the basis of this principle, we can use IR and Raman to elucidate food composition. Besides, we can reduce the size of the spectrometer to make it either portable or handheld sized. So those size of the instrument can be used for on-site or online detection to make the detection to be really user-friendly. So a key part of the spectroscopy analysis is to do the data analysis. So there are a lot of different chemometric models, or we call them multivariate statistical analysis models, that can be used to decipher the spectral features. So among that, we have the conventional chemometrics to apply either the supervised or unsupervised data analysis. In addition, we have the very advanced machine learning models. So for example, the support vector machine or the supervised neural network. So by using either the chemometrics or the machine learning, we can do a lot of analysis for the complicated Raman or IR spectral data analysis. So in my research group, we use different software, for example, Python, MATLAB, and R to do the programming and the coding in order to develop different chemometrics or machine learning models to analyze the spectral features. So I want to use an example to show you a very interesting study about fish authentication by using our portable Raman spectrometer. So before we go that, let me tell you a little bit about the story for this we call it illegal fish laundry. So how we can launder the illegally caught the tuna eventually into the US market. So this map shows the steps in order to do this illegal fish laundry in different corners of the world. So step one, you get a long line fishing vessel and register to a member country. And then you can fish all you can in the high seas without oversight and declare the number of your catch. After that, this is the step we call the transshipment. So you mix your fish in with the legally shipped fish in the high seas. And after that, you ship the catch for processing, for example, in a Southeast Asian country. And after that, you export to the United States as the product of the processing country, for example, Thailand. So this map tells you a classical example regarding the lack of the traceability in our seafood supply chain. So that's why there is a lot of report talking about the fish mislabeling or fish fraud in your local grocery stores. So in this study, we firstly used the conventional method for fish species authentication. So the method is called the DNA barcoding. So we do the extraction of the DNA from the fish samples collected from different resources, followed by the genomic analysis to know the ID of the fish. So as a parallel study, we use our normal method, which is a portable Raman spectrometer, to directly determine the chemical composition of the fish samples. On the basis of that, we try to see if there's a correlation between the Raman data and the DNA barcoding data. So take a look at the DNA barcoding study that we performed in Metro Vancouver. So we collect around 300 fish samples from 3 different sources, including the grocery stores, the restaurants, and also the sushi venues. So let's take a look at the result. So the overall mislabeling rate of the fish samples that we collected in Metro Vancouver is around 25%. Among those, the mislabeling rate is about 28% in the restaurants, 24% in grocery stores, and 22% in the sushi bar. And if you take a close look at the level of the mislabeling for different type of the fish, you will see for some type of the fish, the mislabeling rate is extremely high. For example, snapper is over 90% mislabeling rate. Another example is the yellowtail. So for some type of the fish, the mislabeling rate is relatively low. So for example, tuna and salmon. And among all of those individual mislabeling cases that we determined using the DNA barcoding method, we identify that both the intentional fraud and unintentional mislabeling were observed or identified. So a major challenge for the DNA barcoding method is it is really time consuming and it requires a highly trained personnel to do all the analysis in the lab. So we designed such a portable device, which is called a portable Raman spectrometer. So the size is only, the weight is only about 2 kilograms. And you can see the size is very small. It's comparable to the mouse. So we have 3 different layers of the panel to control the performance of this portable Raman spectrometer. So the bottom layer is to control the performance of the laser. So the middle layer of the panel is to control the performance of the spectrometer. And the top layer is to share the collected Raman spectra through the iCloud and automatically send all of the collected spectra back to our computer in the lab in order to make a matching with the established database by using other fish samples with the Raman spectra. So we carry the portable Raman spectrometer to go to different grocery stores, and you can directly collect the Raman spectra of the fish samples, even including the packed food fish samples in the grocery store. So once the spectra is collected, it will be automatically delivered to the lab. So compared to the conventional DNA barcoding methods, which in total take about 2 days to finish the analysis of the ID of one fish sample, by using the portable Raman spectrometer, we can finish the analysis within 5 minutes for each individual fish sample. So here shows the whole flow chart regarding how we do the experiment. So once we have the fish samples, we use the portable Raman to do the spectral collection, followed by the mathematical model construction, including the chemometrics as well as the machine learning models. I don't want to go to the details for the mathematics, but I just want to highlight the overall accuracy by using our portable Raman spectrometer for fish species identification. So you can see basically the overall accuracy is over 90%. So this means our new method, by using this very small, tiny, and affordable Raman spectrometer for fish species authentication, is highly competitive and reliable. So this is a non-destructive tool and also have the potential application for online monitoring of food processing products. So now let's move on to the second research programs related to food authentication. So this is a nucleic acid-based analysis. And firstly, I want to introduce the conventional nucleic acid method. So this is called PCR. So many of you should be extremely familiar with the PCR method. So in brief, we want to identify the specific type of the animal species that can be identified in the food products. So in this case, we want to identify camel species in food products. So we firstly do the DNA extraction of the food samples, and then we run the PCR to get the PCR applicon. So after that, the PCR applicon will be modified with certain type of the chemicals. For example, the gold nanoparticles were added with the digoxin or biotin tag in order to put all of those elements onto a single piece of the lateral flow assay. So the lateral flow assay is very similar to the one used for the pregnancy test. You have one line, which means your result is negative. You have 2 lines, which means your testing result is positive, because you have a test line and you have the control line. So here shows the result regarding our PCR-based lateral flow immunoassay for the detection of the camel species in the food products. So you can see the sensitivity of this PCR-based lateral flow immunoassay is 100 times higher compared to the conventional gel electrophoresis method to identify the PCR amplicon. So a major question for us is, if you want to run the PCR, we always need to use a bulky and expensive equipment to achieve the PCR amplification. So the equipment is called a thermocycler. It is bulky, it is expensive, therefore it's not very friendly to be used for the on-site or in-field detection. So then we think about if we can use a novel method to replace the conventional PCR method without the reliance of the thermocycler. So the answer is we call the isothermal amplification. So in my research group, we developed two new types of the isothermal amplification for the food authentication study. So one is called loop-mediated isothermal amplification, in short, LAMP, and the other is called recombinase polymerase amplification, in short, RPA. So for the LAMP, the nucleic acid amplification can be achieved at a single temperature between 62 to 66 degrees C within an hour. For the RPA, the nucleic acid amplification can be achieved at a single temperature between 37 to 42 degrees C within 30 minutes. So in this case, we don't need to use the thermocycler anymore. We just need to use a very mini-size water bath to achieve the LAMP reaction. And for the RPA reaction, we can just use our body temperature. You use your hand to hold on the tube or the vacuum containing the whole RPA chemical reactions, and then the RPA can be completed within your hand at the body temperature. So in this case, it maximally facilitates our in-field detection rather than do the conventional PCR detection or study in the lab facility. So one example here I want to show you is how we use the isothermal amplification to achieve a very interesting juice authentication study. And so many of you may have the experience to consume pomegranate juice. So pomegranate juice is, we call it superfruit juice because it has a lot of health benefit to the consumers. It offers a high level of the antioxidants and is highly beneficial to your health. But I'm not sure if you know that or not, that the 100% pure pomegranate juice should be really, really expensive. If you can get a 1 liter of the pomegranate juice labeled as 100% purity, only with, let's say, $5.99 from a local grocery store, I can tell you this is not possible because the price for the 100% pure pomegranate juice should be way more expensive than that. So we do some local survey study to collect a lot of samples that claimed as 100% pure pomegranate juice. By using the molecular method that we designed in my research lab, we identified that those are not 100% pure pomegranate juice as they claimed. So for most of the time, it's a mixture of grape juice and apple juice, because apple juice offer you the sweetness and the grape juice offer you the color. So when you mix the grape juice with the apple juice, it's really similar to the 100% of the pomegranate juice that you can get from the grocery stores. So the whole story for this product is to design a novel method for rapid authentication of 100% pure pomegranate juice. So we use the isothermal amplification method as we presented before. So here shows the data. So we have three different panels of this figure that you can see from here. So panel A, this is the DNA amplification using the primers for the grape, and the panel B is the primers for the apple, and the panel C is the primer for the pomegranate juice. So we see that the primer's design is highly specific that can amplify the certain type of the fruit juice, for example, the apple juice, the grape juice, and also the pomegranate juice. And the whole analysis time for the isothermal amplification of those fruit juice can be finished within 1 hour. So the conventional PCR method always take at least 3 hours. And also we don't need any specific lab equipment such as the thermocycler. A very simple water bath should be enough to achieve the isothermal amplification. So in this case, we can use the isothermal amplification for the rapid authentication of the pomegranate juice. And it is also very useful in the sensors for rapid food safety, quality, and also the authenticity analysis. So now let's move on to the last research topic. So this is about microfluidic lab-on-a-chip platform. So our initial motivation is that there are so many lab components, equipments, and also the personnel in a centralized lab settings. We think about if it is possible to miniaturize each individual component of the lab equipment and also the personnel onto a single piece of the device. So the device that we develop is called a microfluidic lab on a chip. We want all the lab components to be miniaturized and integrated onto a single piece of the small chip. So such a microfluidic device will allow rapid and high throughput screening of different types of the food samples. And in my research group, we developed two types of the microfluidic lab-on-a-chip systems so far. So one type is a polymer-based chip. So this is we call the PDMS, and the other is a paper-based microfluidic chip. So both types of the microfluidic devices are highly cost efficient. So I still want to use the pomegranate juice study as the example to show you how we can integrate the entire isothermal amplification onto a single piece of the microfluidic device. So for this project, we developed a hybrid paper polymer-based microfluidic devices. So in total, there are 7 different layers of this microfluidic device. So we use a technology that's called origami to kind of fold different layers of this microfluidic device so as to achieve the amplification of the isothermal amplification on these microfluidic devices in order to achieve the authentication of the pomegranate juice. So this is the layer by layer view of this hybrid paper polymer based microfluidic devices. So layer 1, this is from layer 1 to layer 4. So for these 4 different layers, they were made of the Waterman number 1 filter paper, except those central area on the layer 4, which is fusion 5 paper, with the diameter of about 5 millimeter for the DNA extraction. So as you can see from the figure, the filter paper was blocked by the wax printing to define the hydrophobic areas, as you can see shown in black color. And this can facilitate the guidance of the liquid wicking. And the layer 5 was consisted of a Y-shaped Fusion 5 paper disc in the center, and also the 3 pieces of the 3 millimeter of the Waterman number 1 filter paper circles in the surroundings. So these paper materials were sandwiched by a thin laminating film. And the layer 6, this is made of a microchip optical adhesive film and 6 pieces of a 3 millimeter Waterman number 1 filter layer of the adhesive films. And the layer 7, this is the layer to achieve the isothermal amplifications. So this is the design for this origami hybrid paper polymer-based microfluidic devices. So what I want to emphasize is that only the A has DNA that can be amplified by the isothermal amplification or the PCR primers, and the DNA will not be able to migrate horizontally on or in the cellulose paper because we use the wax printing to control the hydrophilic and the hydrophobic zones within the microfluidic devices. And then finally, we use a portable charger to achieve the temperature control because, as I mentioned before, the lamp reaction will be only available at the temperature at 60 to 63 degrees C. So the portable charger will be our heating source to facilitate the entire lamp reaction. And eventually, by integrate the isothermal amplification onto this single piece of the microfluidic lab on a chip, we can achieve a very rapid and reliable pomegranate juice authentication within a few minutes. And each test will only cost a few dollars, and this is highly doable even at your home. By finalizing my presentation today, I want to acknowledge some people who work in my lab, especially Dr. Yashi, who did a lot of those authentication study. And I also want to thank my government collaborator, Canadian Food Inspection Agency, to facilitate some research collaboration. And finally, I want to thank the funding agencies, the NSERC, MITEX, and also the Peter Wall Institute for advanced studies. So with that, I want to wrap up my presentation today, and thank you so much for your listening, and I'm open to any question if you have. Thank you so much.